Welcome, everyone. Thank you for joining us tonight. I'm David Dodick. I'm the chair of the American Brain Foundation. And tonight, we have two exciting speakers and an exciting topic. We will learn about a cross-cutting therapy in brain disease, stem cells. Our foundation is committed to sharing valuable resources and increasing public awareness of brain diseases. And our webinar events like this one tonight are opportunities to connect you, our audience, and our followers with experts in various topics of interest around brain disease. The American Brain Foundation funds research across the entire spectrum of brain diseases because we know that brain diseases are interconnected. The underlying biological underpinnings of one disease is similar across many other diseases. So we believe that if we find a cure for one or an advance in the treatment of one, it will have a ripple effect on other diseases as well. And we know that tens of millions of Americans suffer from brain diseases and disorders. In fact, it's recently been estimated that at least 1 in 3 people worldwide are impacted by a brain disease. And that's why the American Brain Foundation has invested over $40 million in research grants to almost 300 researchers. So let's move into our topic of conversation tonight, which is stem cell therapies with our panelists. Tonight, we have Donna Chang, who is the CEO of Hope Biosciences, which is a biopharmaceutical company developing stem cell-based therapeutics for a variety of different conditions, both neurological and non-neurological. And we have Dr. Paul George, who's actually the chair of the American Academy of Neurology Science Committee, and he's also a professor of neurology at Stanford University. So thank you, Donna and Dr. George, for both joining us tonight. Maybe we'll start the panel discussion by having each of you just briefly introduce yourselves. Donna, if you wouldn't mind.
My name is Donna Chang. It's a pleasure to be here. I am currently CEO of Hope Biosciences. We are knee-deep in clinical research, and particularly our main focus is in neurological disease. So I'm happy to be here to share some of that with you.
And Donna, maybe just before we have Dr. George introduce himself, maybe you can tell us your connection to stem cell research, how you got started.
Yeah, well, I've been I did my graduate work in biotechnology, and I was instantly sort of connected to stem cells. I love the idea of being able to regenerate our own bodies, and I really focused on, I ended up wearing many hats within industry and, fell into adult stem cells about 15 years ago, and I've been in it ever since.
Fantastic. Dr. George, welcome. Thank you so much for joining us tonight. If you wouldn't mind just basically introducing yourself and your background and why you're here tonight to discuss stem cells with us.
Yeah, thanks so much, David, and thanks for the ABF for the invite and hosting this event. It's something near and dear to my heart. I'm a stroke doctor at Stanford, and my background is in kind of biomedical engineering and using biomaterials to interact with the nervous system. And my connection to the stem cells really happened during residency and fellowship when I see stroke patients in the hospital. And we've actually made amazing advances in the acute treatment of stroke, but despite that, there's still about 50% of stroke patients are left with some significant disability. And so stem cells are just a really exciting, powerful tool that can help kind of our own regenerative processes after stroke. And so I started a lab that looks at preclinical models of stroke and trying to understand how the stem cells improve recovery and how we can develop treatments from stem cells.
Great, thank you. So maybe I'm going to turn it over to both of you actually and ask you, because there are a lot of preconceived notions about what stem cells are and what they are not. And so it would really be helpful, I think, for this audience to hear from each of you and provide an overview of what we're talking about when we say stem cells.
Sure. Well, I will take a stab at this. I get this question all the time. What is a stem cell? What's the easiest way to explain this? A stem cell is essentially a cell that has no identity. It's a blank slate. It's like a baby can become anything when it grows up. So in this case, a stem cell that has no identity is able to, one, differentiate, meaning transform into various cell types. So different specialized cells. Yeah. And as you can see, brain cells, everything in our bodies, from our brain to our skin, to our hair, to our teeth, every cell that makes us starts with a stem cell. Also, to meet the definition of a stem cell, they also have to have the ability to replicate themselves, so they can make more of themselves or copies. So that's essentially what a stem cell is. Now you can see why these cells are particularly, why they're studied, because if they can make all these cell types, imagine if you have a bad liver and you need new liver cells, then stem cells should be able to become those liver cells. So you can kind of see how these cells could be useful for many applications. But where do you find them? The place where we, historically, the most popular place or the most well-known place is the embryo. And the reason why embryonic stem cells sort of made their entrance and why they are still to this day studied is because essentially an embryo is a big ball of stem cells, right? Every single one of those cells is going to end up becoming an entire human being. So there's essentially no limitation in what they can become. Okay, so they can become a whole human. So with that, there are challenges working with these cells because they are so potent in the fact that they can become, an entire human. There are also other places you can find cells, not just in an embryo. So more recently, you've probably heard of getting stem cells from birth tissues like cord blood, cord tissue, or placenta tissue. All good places to find cells, different kinds of cells, but stem cells are found in those sources. Also, after birth, you can find them in adults. So we are running around with stem cells in our bodies. And if you don't believe me, get a cut and you'll bleed and you'll form a scab and it'll heal itself, right? That is our body's natural ability to be able to fix itself, comes from the innate stem cells that we carry around or are floating around in circulation. There's also a new type of stem cell that's gaining a lot of attraction, which is called an iPS stem cell. Now, iPS cell. Now, iPS is more of a man-made cell. So it's not a source where you go get a cell, but you essentially make it. You take an adult stem cell and you can engineer it, zap it, do different things to the cell to make it embryonic-like, meaning they have all the properties of an embryonic stem cell, but without ever having to come from an embryo. So it's some exciting work that's happening in the iPS world. If we jump into adult stem cells, the only reason why we talk a lot about these is because they're popular all across the board. You hear about adult stem cells all the time because our bodies, again, carry these cells. So most of the time, stem cells are made in our bone marrow. That's the hub for stem cell production. In our bone marrow, there are two major kinds of stem cells. One are called hematopoietic stem cells. They make blood, red blood cells, white blood cells, platelets, that sort of thing. So you might be wondering, okay, well, we're here to talk about the brain. So if we want to make new brain cells, hematopoietic stem cells cannot. Okay, they can only become blood. So how about everything else in the body? Well, it turns out there's another stem cell called a mesenchymal stem cell. These guys basically handle the rest. They are what we call multipotent cells that can become all the lineages that most of our cells, all of our cells come from. So these cells are widely studied and Dr. George can tell you a lot more about them.
Yeah, thanks, Donna. I think just to crystallize maybe a few terms that people hear as they're talking about stem cells, just maybe I'll review a couple of definitions just so when you're reading things in the news or other places, you're familiar with what some of those terms refer to. I like Donna, am just amazed with these cells. I think as she mentioned, the one part of the definition of a stem cell is it can become any cell, or pluripotent is the kind of more scientific word that gets thrown around with that as well. So that it really is quite amazing that from a few cells in an embryo, our whole body develops and they have the potential for all that. So that's the pluripotent nature of stem cells. And then the other part of the definition is that they're self-renewing. So you can imagine it takes quite a few renewal cycles from a couple of cells in an embryo to make a whole human being. So that is the other core component of stem cells as well, that they can self-renew without changing their properties too much until they get the right triggers. So I think those are some parts of the definition. The other terms that you sometimes hear are autologous stem cells versus allogeneic. And I think part of the confusion when I talk to patients or others is they've heard of stem cell transplants for cancer and other things that have been around for a long time. And those kind of fall under the autologous transplants, where it's from the patient's own cells. And it's slightly different from most of what we're talking about tonight, where those are used in cancer, where they deplete the bone marrow and then kind of replete it with the patient's own cells. Most of the therapies we'll talk about tonight are where they take cells either from the patient or from another patient and then use them as a therapy. So autologous are the patient's own cells, and then allogeneic are stem cell types that are taken from another person and then developed as a therapy. A lot of companies make some changes to maybe enhance their qualities and then use them for various neurologic diseases. So just a couple of terms that tough to make out unless you know what they mean. And then just to emphasize, I think it's pretty amazing too that there's these stem cells in the adult body, right? Not only in the bone marrow, but kind of one step down from a stem cell, there are neural progenitor cells, so cells in the brain that can also change into different cell types in the brain. So a glial cell, the neuron, and so these cells also exist or thought to exist in human brains, and in a couple of spots, the hippocampus that makes memories and to make new memories, and then along the ventricles that helps with the olfactory system, but can change course in times of injury. Definitely been shown in animal models. So there are these reservoirs of kind of stem cells in the adult that are interesting for study and for therapies.
So thank you both very much. So Dr. George, can you tell us, you're a stroke specialist, and are these cell therapies being deployed for stroke rehabilitation and stroke treatment? And if so, where's the science there, and where's the clinical application right now?
Yeah, so I think it's an exciting time in stem cell therapies. I view it a little bit like gene therapy, where there was a lot of news and flash at the beginning. It took a little bit to understand how to best use it, and then lo and behold, 20 years later, it's really effective treatments for humans right now, right? Stem cells were the same, right? I think they got their initial buzz in the late 90s, early 2000s, but it's taken us a little bit of time to understand them and figure out how to best use them. In stroke itself, there's over, I think, last time I checked, over 50 clinical trials that are ongoing or completed, and there's two main areas of excitement, or ones that are used. As I think Donna will also talk about, these mesenchymal stem cells are ones that have been used most often, and there's been a couple large trials, one ongoing that these cells are given intravenously and in the acute period after stroke to try to reduce the effects of stroke size. And the thought here is that it's really the regulation of the immune response and some of the things the cells are secreting that will help improve recovery after stroke. And there's been a lot of data in animal models and others that this is the effect. And so that's ongoing and pretty exciting to see the results of that. And then the other is kind of a direct transplantation approach. And I think this has been done by several groups where they near the stroke area or into the stroke area that are transplant stem cells. And initially it was thought that, stem cells, I think early on that they would change into the cell types that were damaged, maybe reconstruct that architecture. But really getting to understand the mechanism of action, a lot of these cells, it's really what they're secreting and how they affect the environment. And there's been now kind of this concept of introducing kind of recovery epochs, or when people have done gene studies of kind of early development when we're forming as young kids, and shown that in times of injury, there's a similar type environment and that stem cells can expand this time window of recovery. And so a lot of these kind of direct transplants or others are looking at that as far as what they're secreting. As the slide shows here, there's a lot of different processes the stem cells have been shown to affect, from helping new synapses form to regulating the inflammation after an injury, to helping form new cells from those body's own adult stem cells that we talked about. So I think that a lot of those are like I said, either in early clinical trials, or there's even a couple late clinical trials in stroke now where there may be some exciting results in the not too distant future.
And we see this slide here these cells have multiple mechanisms of action by which they can improve neurological recovery from stroke or perhaps many other types of injury. It's a complicated slide, but could you walk us through this a little bit?
Yeah, I think it is complicated. I often say I like stem cells because they're smarter than I am, and they can do kind of the reacting to this really complex neural injury environment, right? And so a lot of research has been done, especially on the animal side, try to tease apart these effects. And so one big effect is, again, on the inflammation. There's different parts of the immune system that they can regulate. And inflammation is an interesting one after all types of neural injury, because a lot of it has good effects, right? It can clear out damaged tissue, so new tissue can be remade. But then there's also kind of side effects of that, where the inflammation may get too much and cause more damage. So stem cells can have some role in regulating that. The neurogenesis is, increasing perhaps even new neuron formation, or the neurons that are survive, helping them form new connections. That kind of gets into synaptogenesis, some of the other terms on here. And then the factors they secrete are kind of in a group of been termed trophic factors. And so those are factors that have been collected from the cells that are shown to be beneficial for recovery. And some of the research now is looking at exosomes, or particles that these stem cells release to try to improve recovery even without the cells. Maybe you can make these cells secrete something in a lab, and then you actually just give the patients what those cells have secreted to improve recovery. So I think the better we understand the mechanisms, the more refined treatments we can develop.
Thank you. So Donna, your group has done remarkable work actually in growing these cells, and it perfected the art of delivery and volume, number of cells. It's complicated, but maybe you could walk us through that a bit, as well as tell us what the landscape looks like right now about the treatment of other neurological diseases beyond stroke, and perhaps other diseases in general.
Sure. We started Hope Biosciences about 7.5 years ago, really to try and come up with a solution for why stem cell therapies are not approved in the United States. That I'm sure many of you will have questions about. Why can we not access these treatments? It's because there's failure when these cells, when these treatments get to larger trials and late stage trials. And what we think the pain points in developing good, effective cell therapies really come down to mode of delivery, how many cells are being delivered, the purity of that product, the viability, meaning viability is just, are they alive or not? You need, dead. I always say it doesn't matter how many cells you give someone. If you give someone 1 billion dead cells, it isn't going to do anything. And that sounds so funny, but the reality is the field, we have a lot of issues in the field to try and figure out. So that's really what Hope Bio was set on a mission to do, not to really look at disease particularly, but first to figure out, can we even find the right cells? Can we make adequate quantities? Can we make them fresh so that we know are viable? And can we do this repeatedly? So, I did a quick search on clinicaltrials.gov before, and that's a website where you can go see all of the latest clinical research up to date, all the protocols that are happening globally. And I did a search today, and it came out to 801 stem cell studies, specifically in neurological conditions or disorders. So that's a lot. About 150 of those are currently active. And if you look at all of the trials and you kind of break down where we are as a field, what I have here highlighted in this red is kind of a summary of where we are. For various conditions, they'll use autologous or allo cells, meaning your own, as Dr. George told us about using your own. You're the donor for yourself, or you get a donated stem cell and given that. That you have different cell sources. They're given normally a single dose. I've seen some protocols giving 2 doses, but mostly 1 or 2. And each dose is about 60 to 120 million. And it actually, most of the time, is dosed per weight of the patient. And this is all important only because we're talking about, a dose of medicine. That's how you have to look at this. So this is the dose that's currently given. And it's being delivered either directly to the injured site. As Dr. George mentioned, you can do it directly into the brain to get it near that stroke site, or you can give it intravenously. That's through the bloodstream so that the cells figure out where they need to go. But most products are delivered frozen, and that's done because of logistics. So if stem cell products are thawed and then administered, we have to ask, how many of those cells are actually alive and well and are going to actually work at fixing the problem. Most of the trials are in phase 1 and 2, which are sort of early stage trials. So here's what we are with some of the applications at Hope Bio. The most advanced, I would say right now, is traumatic brain injury, because it was the first one that we started in neurology. And you can already see the amount of doses is different. So the order of magnitude is changing. If you see, we're now giving 3 doses of cells. We're doing this intravenously, and we're giving them through the bloodstream, and yet we are finding changes in the brain. Not just structural changes, but even changes in inflammation in the brain, which is a really just an incredible function of these cells. And if you remember the slide that Dr. George showed with all the different ways MSCs work, that's our headache that we have to deal with. What if you had a drug that could do 10 different things? How do you figure out which pathway they took to solve the problem? So that's our challenge we have as we're trying to get this drug out to people. But yeah, right now we are giving lots of cells. And if you can see our portfolio of other neurological conditions we're studying, the numbers are getting bigger and bigger. And the reason is complex diseases like spinal cord injury. In order to repair an injured site, if it's a complete injury, for instance, it requires lots of cells. And so we're really on just at the tip of the iceberg here when it comes to the application, but we do see that stem cells are going to be it's a ongoing treatment. It's not like a moonshot. You get one thing and it's going to fix you. So if a doctor is trying to sell you one treatment is going to fix you, it's probably not going to be the case. If you think of degenerative disease, we are constantly degenerating. So you have to regenerate in the same way you degenerate. So it will require the re-administration of cells. And so can we do that safely over time? That's sort of what we're studying right now. So you can kind of see where we are. Yeah.
So thank you, Donna. When I look at the sample size of these trials, it's relatively small, right? So there are a lot of people out there with multiple sclerosis, with TBI, Parkinson's disease, Alzheimer's disease. And you could say, well, they're interested in stem cell therapy. A lot of people are asking about, what about stem cells? And, we could say, well, let's search for a clinical trial that you might be eligible for. But what if they're not eligible? And what if there are no open trials for them to participate in? They take it upon themselves to try to find a place where they can get stem cells. So can you talk to us about the safety, the wisdom of doing so? Because there's a lot of people out there, perhaps some people watching this, tuning in tonight, or in the future when this is posted, who are going to want to know, where should they go? What should they do if they only have one shot at this and they want to take advantage of cell therapy?
Dr. George, you want to take a stab at that?
Sure. Yeah, no, it comes up quite often, right? Unfortunately, with a lot of our neurologic diseases, there's not really curative measures at this point, right? And so stem cells are one that have some hope that hopefully in the future we can offer some of that. But I think the safety risk outweigh any benefit, of trying to get these cells outside of a clinical trial. I start off some of my lectures with just sad cases of people who went to other countries and received therapies that may not have been as overseen as they are in clinical trials or other things and have bad outcomes. So I tell my patients really, hopefully you can qualify for one of these clinical trials if you want it, because in the US, that's really the only way to get it for neurologic diseases at this point.
Yeah.
I would strongly recommend trying to find a trial site. Yes, I understand qualification is always a problem. There are other avenues that manufacturers can be contacted directly for different pathways. There are different FDA pathways that allow accessibility, like expanded access. As you can see here on spinal cord injury, you'll see the word expanded access. That is essentially a clinical trial for one person. So yeah, you don't meet a criteria. First of all, spinal cord injury is all different. So it's very hard to find 5 people that are exactly the same injured site, same severity. So this is the reason why we do this under expanded access. But manufacturers are, I think, more inclined to releasing their products under these programs. There's also Right to Try, which allows people with terminal conditions to have access to investigational drugs. So they have to be going through a clinical trial and they have to have passed safety in order to qualify for that. But that is also another avenue that patients can ask manufacturers directly. I would strongly encourage you to find someone in the United States to ask for that access. And the only reason why I say this is because what you get overseas, you can't qualify. Rarely are you given a certificate of what the product is or know what exactly it is, how it's been tested. And all of these countries don't have the same standards as are here in the United States. And so that's why we would strongly recommend trying to find a clinical trial. And if not, that's the drug that you really want, to contact the manufacturer and see if there's any way that you can participate in another program.
Thank you. Thank you both for that. I'm going to open it up for questions, feel free to type any questions you have into the chat, or open your mic and feel free, or raise your hand and feel free to ask a question. But I don't see any right now, so I'm going to ask another one myself, which is, you mentioned safety. What do people have to be concerned about from a safety standpoint? And I imagine, although you can correct me, that the safety differs whether you're receiving autologous, which is your cells, versus allogeneic, which is another person's cells. So maybe you could tackle the issue of safety, side effects, short-term and long-term, actually, if we know.
Yeah, from safety, we've done a lot of clinical work in a lot of different patients, a lot of different conditions. So across the board, when you talk about like you read papers about safety of autologous cells, they will always talk about, well, if you get your own cells, it's perfectly safe. Yes, the idea of you receiving your own cells, in that your cells, you sitting here right now with your cells, is safe, is correct. But once the cells leave your body, okay, they have been removed from your body, and then they are processed for a certain amount of time. There are lots of things that can happen during that time. You can get contamination. There can be mutations that occur with the cells. It happens all the time. These aren't like rare occurrences. They also like to differentiate in the lab. So you want the body to tell the cells what to become, but they can actually transform in the lab. And so you don't want, if you're trying to receive stem cells, but you get a bunch of skin cells and stem cells, that's not going to be a pure product. So you really need to, safety. And I think the reason why FDA says, we want to regulate the safety of these products is really not so much the worry of the cells that are coming out of your body, but really what happens during the time they're outside of your body. Hope that makes sense.
Yeah, it does.
And I think just the overwhelming results of these trials is that in the proper setting with the proper regulation, they are very well tolerated and safe. I think that's even these small trials and bigger ones, there really haven't been a large number of adverse effects. So I think in the right setting with the right oversight, they have been shown to be very safe. We obviously think about things like tumor, right? Putting a cell in that can become any cell type. We monitor closely for that. That really hasn't been shown to bear out. Obviously, we haven't monitored someone for 50 years or something, right, after these transplants. But so far, in animals and humans, that it's monitored closely. We haven't seen that. When you're injecting cells, especially into the brain, sometimes you think of seizures. Those can occur on rare occasions, but across all the trials that Donna had mentioned, it's really not seen that widely. So I think, again, most of the trials have shown that they're safe, and proper monitoring for these side effects is pretty good.
Yeah. Marcus Wolf, you have a question, sir.
Hi, yes. I'm a gerontologist, and I'm working on memory cognition and slowing among older adults. And I remember someone mentioning that the stem cells are more of an ongoing. How do you think that would affect, or what do you think is is there a possibility that the stem cells could help slow memory help with memory cognition and slowing as opposed to waiting until someone gets to a point?
It's a fantastic question. And I think that's where we're all headed to, right now in when we're looking at the disease. So if this is the end stage of the disease, what is end stage? Particularly, let's say in Alzheimer's disease, I think Dr. Dodick can verify this, but Alzheimer's disease, you don't just wake up and have it. It takes decades, 20 to 30 years estimated, that this disease is slowly causing this degeneration. So at some point you're going to see your first symptom, but it's already been decades since you've had the condition. So really, we want to be able to treat this sooner than later. Now, everything is easier to treat if you treat it sooner than later. So if we can get cells delivered sooner, which is, I think, why you see more and more trials trying to tackle disease that's more mild. They're trying to stay away from the more end stage, and the reason is because it's very difficult. It's a very difficult, challenging population to work with. So they're trying to design these trials a little bit sooner. So I think what you are thinking is logical, and the field is going to move that way for sure.
So at some point, I put in the chat, we're thinking of well care and preventative care. At some point, it could possibly be preventative, because there's a lot of things on the market now, Prevagen and all these other things that claim to do. I'm not going to get into that, but I'm just wondering, do you think at some point it could be preventative?
Yeah, well, I definitely think that that's where it's going. Yeah.
And I think there's starting to be some understanding too. It's a very complicated question, as you kind of indicated, and Donna, there's so many steps to understanding the disease itself, to understanding what stem cells can do for it. We are starting to understand kind of the body's own mechanisms of repair. And so, yeah, I mentioned those endogenous stem cells in the brain. We know those decrease over time, and stem cells have shown some effect with increasing those numbers, right? So I think there are some mechanisms we're beginning to understand that could help us develop those therapies. But yeah, there's a lot of steps in that pathway as far as when to deliver them, how to diagnose the disease, and which stem cell therapy would be most effective for that.
Dr. George, Bob has a question here, which is, do you know if stem cell therapies could be beneficial for his rare condition, idiopathic hypertrophic olivary degeneration?
Yeah, these rare conditions are always tough, right? Because we don't have big studies to guide us. I think, in some sense, even though it's not really low lying, stroke is a nice one because we know when the insult happens, and then we know where the territory is damaged. I think a lot of these degenerative diseases get back to the previous question, right? What's the mechanism of the disease? A lot of them we don't understand. I think it's a reasonable approach to consider, right? As I said earlier, sometimes the cells are smarter than us. Maybe in the right environment, they respond in ways we don't quite understand yet. But yeah, I think it just hasn't really been studied yet. But in theory, I think there's some possible ways.
Dr. Schubenstein.
Hi, and thank you both for the time. This is a fascinating area of research. My question is, given the exciting work going on in stem cells, and at the same time, a lot of really exciting work going on with hyperbaric oxygen therapy, are either of you aware of anybody doing combinatorial research and looking at the two together?
It should definitely be studied. I am not aware of any, institutional, level work being done in that area, but it should definitely be looked at because, ultimately hyperbaric, along with a lot of other, a lot of functional medicine physicians are also looking at, these tools are being used to sort of normalize or create homeostasis in the body. And that's really important to look at. If someone's eating the right diet, if someone's doing the right things, taking care of themselves, it's a good environment for cells, even for application of any drug for that matter, right? You want to create that environment. So it makes a lot of sense that studying those things together. Yeah.
Yeah, and I think it's a great question and gets to a point of, optimizing the stem cell effect. In our lab, we combine electrical stimulation of the brain with the stem cells and kind of getting to that point that perhaps what the stem cells are secreting is creating kind of a younger brain environment that is more accepting of remodeling or other things. And then you can guide it with electrical stimulation or other modalities. So I think it's a great question, and agree that it's an area that's interesting to look at.
Great, thank you both.
There's a question here from Dr. Alan Purdy from Canada, who asks about, and maybe this one's for you, Dr. George. He asks about a physician from or scientist from Stanford who is developing organoids, clumps of neural tissue in a model of producing a muscle twitch. So he's always skeptical and wonders about your thoughts on this type of research, if you're aware of it. Claims to be reverse engineering a brain. Any thoughts on that?
Yeah, I know Sergey well. I think he's one of a number of groups that are using the induced pluripotent stem cells, so those iPSC cells, where they can take fibroblasts or skin cells from patients and then give the right stimulus to bring them back to kind of a stem cell state, like Donna was talking about. And from that, they can grow kind of these mini brains or organoids in culture by allowing the cells to grow over months. And it's really interesting, actually. They've done looking at markers in a kind of from a child in the womb, basically, right? Markers of RNA-seq, and if the cells are in a culture dish for like 3 months, they have the same markers of a baby in the womb for 3 months. So they've kind of done some of this tracking that they form in similar ways to the brain, obviously a different environment. And then, Dr. Pascal has really pioneered combining different parts of the nervous system together from these organoids. Like you said, like you talked about, you can go from the neurons themselves all the way to a muscle organoid and make it activate. So I think there's a lot of caveats, right? It's not like he's going to create a whole brain and nervous system, but I think it gives a unique opportunity to study different disorders, because they can come from patients who have genetic disorders, and then you're making a model of a brain in a dish that can perhaps give us more insights than just regular cells could into that disorder. And there have been some moves to even make those a little therapeutic, where you would transplant. He has one paper where they transplanted that into a rodent brain and improve some recovery. But I think that's further out. But I do think they're powerful tools to study diseases, because they come from patients and can form more of a culture system than just kind of a 2D layer of cells on a dish, right? It's a little more like the brain to study.
Yeah, interesting. There's a question here. Are there companies that promote storing individual stem cells for future use? So Dr. Chang, you'd be the perfect person to answer that. And when you're answering that, could you also tell us what type of tissue do you use to grow these mesenchymal stem cells, and how long does it take? Why store them? Why not just get them when you need them? Even your own cells, how long does it take to grow them?
Sure. So I want to say a word about banking, because I think the general topic of banking, I hear it more and more, and there are companies that are popping up and coming out of the woodwork saying, we bank stem cells, and probably you've heard of cord blood banking. This has been long time referred to as stem cell banking. Well, if you remember the two types of stem cells, cord blood contains something called hematopoietic stem cells. Okay, so if you're hoping that you can use these cells one day to fix your brain, that is not going to be a reality. So you have to figure out what stem cell is being banked and also how it's being banked. So if you take a tissue or blood and you collect that and you put it in a freezer, then what happens is 20 years later, you need cells. They're going to take it from that freezer and they're going to thaw it out. And most likely a lot of it will be unusable. They'll give it to people like us and say, please make something out of this. And we can't. There's nothing in there that is viable. So, banking, if you want to bank, you have to first prepare the sample so that it can be banked. And there are many ways you can find stem cells, right? All the different sources we talked about. But in adults, pretty much every tissue of your body has these cells. We at Hope Bio, we use fat only because fat is a very easy source to work with, and everyone's very excited to give us some fat. So we gladly take it. And from 1 tablespoon of fat, we can then find the MSCs. So remember those mesenchymal stem cells, the ones that can do all the things we are likely going to need them to do later on in life. We will collect those cells, and only those cells. We'll get rid of everything else. So all the fat, all the tissue gets removed, and we only use those cells, and then we grow them. We culture them. So we make copies of these cells over and over and over again until you have an order of magnitude that makes sense, that can survive freezing, thawing, making more cells. And so that's the logic. We really want to bank something that can be used over and over and over and over again in the future. So you bank once and you'll have cells for the rest of your life. So that's how we do it. Yeah.
And I would imagine that, how long does it take to actually grow them? So if someone has a spinal cord injury and they haven't banked cells, how long would it take to grow those mesenchymal cells?
So we'll get them out in 10 days. So if you're already banked, we'll get them out in 10 days. If you are not banked, then it will take 2 months to bank, because it takes us that long to prepare your cells to cryopreserve them for future use. And from that 1 tablespoon of fat, we can make over 414 billion cells. So that's over 1,063 treatments of 200 million cells for the use for the rest of your life.
And if you took 1 billion cells out of the freezer, what proportion of them are going to be viable?
Yeah, it's a great question. The problem with that is that it varies greatly. It can be anywhere from 10 to 80%. It's such a huge range. And you can freeze 2 vials exactly the same way and thaw them out, and one can have a totally different viability than the other. So, when working with these biological products, they're live cells, they have personalities of their own. They are, it's not just about their viability. It's about, are they going to be able to be, are they alive and are they functional? So they got to be both, because they got to work. So they may look viable in a microscope. So you might be able to count them and say, oh, it has 80% viability. But when we actually test them to see what their function is no good. That happens all the time. So you really want to make sure that the cells that are given to you at the end of the day are tested to ensure that they are not only the identity that they are, the purity, but also have the function that you want them to have, which is potency.
Yes. Yeah. This is complicated.
It is. So if anyone is trying to bank cells, and if they're telling you we're going to take this and stick it in a freezer, you can save your money. You really do need to have enough cells to be able to recover later on for treatment. Yeah.
Yeah, I know, and it really is one of the maybe not bottleneck, but one of the challenges, right, for developing these cell therapies is how to produce enough cells to be meaningful and applicable to the patient when they need it. In California, I know they're thinking of how to develop centers that, for the iPSC cells especially, could rapidly take skin cells and then produce a cell type that you need, so that if you didn't biobank, or for more widespread use for people who don't have access to biobanks, etc., there would be a kind of a systematic way to make sure it's the right differentiation state, the right cell type, and a more standardized process. Because especially with like the embryonic stem cells or others, it's amazing the differences using the techniques if you start to expand them for long periods of time and things like that. So some people think that the iPSCs have some advantage, that you could use the same technique in a shorter period of time and have a similar cell product for therapy. So there's a lot of different angles people are looking at to get a cell product to patients.
Dr. George, are there any trials, there's a question here, in epilepsy?
A little out of my domain. I don't know, no large scale ones. I think there, in reviewing literature and things, there are some small scale trials that have been done, but I'll confess I have not been keeping up with that literature as much.
Yeah, not in the United States, but there are several that are being done overseas. And in fact, we have a nonprofit research foundation that does a lot of these compassionate use and expanded access cases. And FDA did approve our first pediatric case of drug-resistant epilepsy in a, I think he's 11 years old. So it's the first case that we're doing, but we had observed in our traumatic brain injury trial, we had observed several cases of patients that had seizures that really got better after treatment for their TBI. So that is what led us to the application for epilepsy. Yeah.
There's Marcus Wolf, you have another question. Is it better to use infant stem cells in treatment? So I think you might have addressed that, but maybe you want to just go over that again, Donna?
Sorry, what was that?
The question is it better to use infant stem cells?
Yeah. It's a great question because we in theory think that, okay, if you're an infant, you have more cells. So yes, if you take an infant versus an adult, the infant will always win. They do have more cells. I mean, they're a giant ball of stem cells if you think about it. But again, when you're receiving stem cells, if it's coming directly from the source, you're looking at it like one-to-one, then yeah, that would be the case. There is a superiority in that sense. But if you are removing the cells and you are making adequate doses, now you're making a therapeutic dose and you are making many of those, then now you're getting more over time. Therefore, it probably, it's not apples to apples. It's not apples to oranges, right? You're looking at it from a numbers perspective. You always have to look at it from a dosage perspective, as well as, how many administrations you're receiving. Each dose. Yeah. So I think the answer is our cells, if given enough, are just as superior or are able to fix the problem, depending on, of course, what condition it is. But yeah, our cells are perfectly good. Yeah.
Mm-hmm. Dr. Schubenstein, I can't tell whether you raised your hand again or it's still up.
No, I had another question. If I may.
Okay, go ahead.
When I speak with researchers in this area and when I read the papers, something that I find that I struggle with is the way the cells are produced, quality controlled, and then the dosing. Really importantly, the dosing is wildly different in different trials for the same conditions. And so I'm personally finding it very hard to understand signal from noise in the data, given this really diverse set of approaches to production and dosing. So I was hoping if you guys wouldn't mind talking about what's your perspective on that, since you both do so much of it and I'm sure see what the other people are doing. How do you think about that, and how should we think about that as we think about doing more in this area?
Yeah, I'm happy to take a first stab, Donna, and feel free to fill in the blanks here. But yeah, it's a great question. I think just like the terminology has been a little messy with what is a mesenchymal stem cell and other things, understanding what each cell product is often difficult and can have a big impact on what number of cells are effective. And then I think confusing even further is there's various types of delivery, right? We talked about intravenous into the blood, injections into the brain. People give them intranasally, right? Mm-hmm. So there's a lot of different ways that people are delivering these. So probably the method of delivery also is a big factor on how many cells you need. So I think what's important is that as much as we can understand in preclinical models is what dose is needed to have some effect, and then early clinical trials with dose escalation, things like that, to understand for a specific disorder or cell type what is needed. That's the research that needs to be done when you get to the really big treatment trials. So that's kind of how I view it, kind of looking at how that cell line was developed, what data there is for each step of the way, either animal models to early human models, and onward, because it does get confusing. Again, different way, all these different variables.
Yeah.
Sorry, Donna, please go ahead.
Yeah, I think a good way to kind of like filter through any trial is, yes, to try and understand what is the product, and most of the time you can get an idea. You'll definitely be able to know whether it's autologous or allo. You'll be able to, hopefully they'll be able to tell you whether it's fresh or not. And with that information, then you can already kind of, and with how many cells they're giving, if you have that information, you can pretty much guess or predict what the outcome is going to be. And I say that only because if the population is not identified, if the QC hasn't been done, let's say, to measure what the cell markers are, to tell us what actual stem cell this is, then I wouldn't even consider that paper, only because there's no way to validate that what was given to the patient was actually stem cells. And also, I would also try to, if we're trying to study what the effect of a stem cell is and try to validate that these stem cells are actually going to work, then we should stay also away from mixed populations for now. We should look at individual populations and then study whether you mix them if they're better. All right, because that takes out some of that noise. So that's how I kind of filter through studies. It helps me sort of look at what's working and what isn't. Yeah.
Great question and great answers. I think we have time for one more question, and there's two questions. Well, there's a number of other questions. We can't get to them all, but there was a question about burns and there was a question about rheumatoid arthritis. So my question is, maybe you could address both of those entities, as well as earlier, Dr. George made a comment about delivering cells into the diseased organ, like the brain. And you talked, Donna, about sometimes giving them intravenously, and they find out where they need to go. So for something like arthritis, for example, or for something like brain disease, do you have to inject it into the brain or into a knee? Or can you just give it IV and they find their way there and they cross the blood-brain barrier, or they cross the synovial barrier and get into the joint? So can you tell us about burns and rheumatoid arthritis, and then in general, how best to deliver these cells, and how do they find their way to where they need to be?
Okay, so burns, yes. These cells can heal burns. In fact, we do a lot of burn research as well. It's a great application. I think it'll be used more. RA, our very first clinical trial was actually RA. People ask me, why did you study RA? Out of all diseases, why RA? Because, for us, we thought it's the easiest. And it's the easiest only because, in our mind, because these cells are, again, powerful anti-inflammatory. They excrete powerful anti-inflammatory compounds that help attenuate the inflammation, which is systemic in rheumatoid arthritis. So we're really depending on that. And if we give it intravenously, it does really do a great job. We published our first study in that, where all 15 patients, we saw exactly the same result. But it was because they all received the same product. So again, you go back to dose and you know what the cells are, and they all receive the same amount. So that's how you're able to get a consistent effect, in my opinion. I think that's what is very important to get. For delivery, it's a numbers game, right? So if you have a small amount of cells, let's say you're a research scientist, you made a very small amount of cells and you want to make sure you get them to the specific area, then yes, you're going to inject it directly into that area. Because when you give an IV, you're already factoring that probably 90% of cells are going to be lost elsewhere. Okay, because they're going to go into the bloodstream, they're going to go into your lungs, they're going to get into your heart, and they're just going to go wherever they need to go, and they'll be very distracted because our bodies are degenerating everywhere all at the same time, irregardless of disease, right? Aging sort of like a disease. It's a terminal disease, right? So we're constantly degenerating. So we need to regenerate, and that's why our cells are sort of distracted in that way. So there's a small homing percentage. So if we're talking about the brain, approximately 10% of cells will make it up to the brain, depending on the condition. It also depends on whether the condition is acute or chronic. We've observed differences in that as well. So I think, yeah, the delivery, so the reason why we go IV is because we have a lot of cells and we can make a lot of cells. So we take the liberty of going the IV route.
Yeah.
That's just a couple of things on top of that. Great answer. I think these cells do have a really inherent ability to home. So a lot of studies have shown when there's injury, these stem cells really find that injury and can navigate through blood vessels, through tissue to get to the area of injury. So that's really one benefit, I think, of having different, being able to deliver in different ways. And then, even IV, if it's not the site of the injury, there's been a lot of studies, Sean Savitz, kind of in the neural realm, others have shown that they have a great effect on the whole body's immune system, right? There's actually dramatic changes in spleen size, other, thymus, other areas that kind of regulate the immune system. So they do have this powerful impact kind of systemically on inflammation or other processes.
Well, you know what? I can't thank you both enough tonight. I mean, this was superb. Great presentation from both of you. The answers to really good questions are just terrific. It's a hot area. It's an exciting area. Patients are asking about it all the time. So I hope that everybody found this useful. I certainly did. And I thank you both very much for coming. And before I say goodbye, I want to thank you all for joining us tonight. As you know, we host free webinars every month, and we'd love to see you again. Next month on November 14th, we'll host a webinar on cross-cutting therapies in brain disease, focusing on focused ultrasound with Dr. Andrei Alexandrov and Dr. Neil Kessel. So you can also follow us on social media to stay up to date on brain disease information and research and on upcoming webinars. And you can also support some of the innovative research that you heard about tonight, as well as other research that the American Brain Foundation supports. So no donation is too small, and we're deeply appreciative of all of you tonight who have given already to the American Brain Foundation. So thank you all very much. And thank you again, Dr. George and Donna, very much for your presentations tonight and for sharing your expertise. Have a great night, everybody.
Thank you.
Thank you.